Okay, picking back up with the power plant of the cell. So we just finished talking about a bunch of different organelles that do a bunch of different stuff. And we had mentioned how food is brought into the cell in a food vacuole, and then a lysozymes fuse to the food vacuole and release digestive enzymes and acid and break it down. Well, one of the main things you want to get out of that is glucose, because glucose is used in the production of adenosine triphosphate, ATP. So this production of ATP occurs in the mitochondria. So the mitochondria uses sugar, ideally glucose, and makes ATP. And the process in which you make ATP is referred to as metabolism, also referred to as cellular respiration. So that is using glucose to create ATP. Here is a mitochondrion. A mitochondria is another double membrane structure. So here's an outer membrane, and then here's an inner membrane. In addition, the mitochondria has some neat features going on. One of them being that there's DNA inside a mitochondria. A mitochondria has its own DNA. Furthermore, as you look at it, you can see that DNA is circular DNA. Mitochondria also have their own ribosomes, which means they can produce their own proteins. That inner membrane is filled with a fluid called the mitochondrial matrix. So I guess that's where you fight agents, when you're inside the mitochondria. Can't help it. I gotta make matrix jokes. It shows how old I am, but it's funny to me, and that's all I care about. Anyway, so you've got the outer membrane, then this space called the intermembrane space, then you've got the inner membrane. Inside the inner membrane is the mitochondrial matrix, and then in there you also have DNA and ribosomes. Pretty funky that you'd have an organelle with DNA and ribosomes, don't you think? Pretty weird. Okay, so physical features. Another important thing to mention is the cristae. Cristae are folds in the inner membrane. They serve to increase the surface area available for ATP production. Basically, if you were to think about this as sort of lines that you could unfold, so if you were to say cut right here and then grab on either side of that cut and just peel it like that, these folds mean you're going to get a lot more when you peel it out. When you straighten it out, there's going to be a whole lot more to it than if it were just a smooth, oblong spherical structure like the outer membrane. So the inner membrane provides a lot more surface area for ATP production. So that's where respiration proteins are found. Provides increased surface area, helps speed up metabolism within the mitochondria. Then you have the mitochondrial matrix, which is within the inner membrane, and it contains enzymes that speed reactions for respiration, making ATP. It has mitochondrial DNA, abbreviated mtDNA, which is the instructions for building the mitochondria, and that's not from the DNA in the nucleus. In fact, the mitochondrial DNA is all on its own. The mitochondrial DNA I have in my mitochondria, in my own cells, is identical to the mitochondrial DNA in my mother's cells. It is the mother who passes mitochondria to her offspring. That means, should I ever have kids, the mitochondria in my kids will not carry my DNA. They'll carry Crystal's mitochondrial DNA, were we ever to have kids. So that's kind of neat. You can trace back mitochondrial lineage from mothers to mothers, and the only differences you get in the mitochondria are from random mutations, and those don't tend to happen all that often. Mitochondria tend to be stable. In fact, they're stable enough that a lot of scientists will do mitochondrial DNA comparisons of different organisms to try and get an idea of how closely related they are, evolutionarily speaking. Pretty cool. Okay, so we have enzymes. We have mitochondrial DNA, which is circular DNA. Then we have ribosomes that produce the enzymes used in cellular respiration. Pretty neat. All right. So mitochondria, all about making ATP, using glucose to make ATP. All right. Well, plants and certain photosynthetic other organisms can do a pretty neat trick, and that's use sunlight energy in the production of their own glucose. So the chloroplast is the organelle where photosynthesis occurs. Photosynthesis is using carbon dioxide, water, and energy from light in order to make glucose and oxygen. So the organelle in which photosynthesis occurs is the chloroplast. So the chloroplast's job is to absorb a bunch of sunlight and use carbon dioxide and water and then energy from sunlight to convert that into glucose and oxygen. So plants have that nice little setup because they can make their own glucose and then send that glucose to their mitochondria to be used up for making energy. Pretty sweet. So chloroplasts are the site of photosynthesis. Their physical features, they have, similar to the mitochondria, an inner and an outer membrane and an intermembrane space between them. There's also the stroma. The stroma in a chloroplast is the liquid filling the inner membrane. So mitochondrial matrix in the mitochondria, stroma in the chloroplast. Then we have thylakoids. Thylakoids are rather interesting. They're a membrane, and they look kind of like a little poker chip, like a little green poker chip surrounded by a membrane. And in fact, inside that little green poker chip is a hollow cavity. And inside that hollow cavity are the molecules responsible for absorbing sunlight. So you remember that from Chapter 7. Or you'll read about that in Chapter 7, depending on whether or not you're out of order. So they're little green disc-like membranous sacs. Look kind of like individual poker chips. Now, just like poker chips, thylakoids can be stacked up, one after the other. And when you stack a bunch of thylakoids up, you get one stack called a granum. A granum is a stack of thylakoids. Multiple stacks of thylakoids are called grana. Just knock off the UM and put an A. So a granum is a stack of thylakoids. And you can see their membrane, so there's a membrane around them, and so all these thylakoids are connected together to each other in a stack, and then the stacks are connected to each other as well. So pretty cool. There's the outer membrane, there's the intermembrane space, and then there's the inner membrane. Then we have the stroma filling it up. You have individual thylakoids, the poker chips. Poker chips are stacked into a granum. Multiple grana are connected together by that thylakoid membrane. Only organelle in the entire animal kingdom where you find three separate membranes, an outer, an inner, and the thylakoid membrane. Kind of cool. Okay, so in the stroma, we have the enzymes responsible for speeding up the reactions involved in photosynthesis. We also have DNA. Chloroplasts have their own DNA. That's circular. And then chloroplasts also have their own ribosomes that are involved in making the enzymes for speeding photosynthesis. So why am I emphasizing the fact that mitochondria have circular DNA, enzymes, and ribosomes, And chloroplasts have enzymes, circular DNA, and ribosomes. Well, let's think back to, one lecture video ago at the very beginning when we talked about the two basic kinds of life. What were the differences between prokaryotes and eukaryotes? Specifically DNA. That's right. The prokaryotes have circular DNA. So chloroplasts and mitochondria have loose circular DNA. They have their own ribosomes. They're very small in comparison to the cell around them. And even more interesting, they replicate on their own. When it comes time for a single cell to divide and become two cells, it's not the mitochondria aren't replicated as a part of this process. Rather, the mitochondria will duplicate themselves on their own. They replicate separately from the rest of the cell. So that's pretty crazy. They have their own DNA, they have their own ribosomes, they're very small, and they replicate on their very own. If you didn't know any better, if I didn't call them chloroplasts and mitochondria there, and not that you can see me covering up chloroplasts and mitochondria with my thumbs, but if you didn't see these two words and I just said, here's a cell that has loose circular DNA, has ribosomes, very small, and can replicate on its own, what would you tell me? You'd tell me that's a prokaryotic cell. Well, guess what? Endosymbiont theory. Endosymbiont theory, by the way, it's a scientific theory, so it's very well supported. Endosymbiont theory states that the chloroplast and the mitochondria were once free-living prokaryotes. That's pretty crazy when you think about it. How cool is that? That they were once living on their own. At some point, some ancestral cell of ours ended up swallowing up that cell, taking it into it would have been big, so a food vacuole. And for some reason, it didn't digest the target. Instead, they formed a symbiotic relationship in which the larger ancestral proto-eukaryote provided nutrients and protection to that little proto-mitochondria or proto-chloroplast. And in return, the proto-mitochondria provided a bunch of ATP, or the proto-chloroplast made glucose for the cell. Either way, turned out to work well for both of them. So it's pretty cool. Studying mitochondrial or chloroplast DNA, we've been able to show that they're very closely related to certain free-living prokaryotes. So that's why endosymbiont theory has gone all the way to the point of being a theory. It is extremely, extremely well supported. Pretty awesome to think that every cell in your body is the product of an extremely lucky symbiosis that occurred once upon a time in time immemorial. Okay, we're actually done with organelles. And so I want to get into the last sort of complex concept here, and that's the concept of the cytoskeleton. The cytoskeleton is not an organelle. Rather, the cytoskeleton serves a similar purpose to our own skeleton. Our own skeleton provides us structural support. It allows us to maintain our shape. It can help us move. Well, the cytoskeleton is this microscopic network of little structures like filaments and tubules that connect to each other, and they help support the cell. So why doesn't everything in a cell just slosh around? Because of the cytoskeleton. The organelles tend to be anchored in place in the cell because of the cytoskeleton. In addition, the cytoskeleton maintains the cell's shape, and it's just overall really cool. It is made up of two basic proteins. proteins called microtubules and proteins called microfilaments. Microtubules are larger than microfilaments. Microtubules are tiny, hollow, tube-shaped proteins, and they form a scaffolding to support the shape of the cell, and they also form this sort of track on which vesicles and organelles move. So it aids in the movement of vesicles or organelles to where they need to be. And then you have the microfilaments, which are teeny tiny solid filaments. They're not tubes, they're solid. They have some other interesting things. They carry out fine cellular movements, including contraction and cytokinesis. Cytokinesis is the movement of the membrane. So, for instance, formation of vesicles would be a form of cytokinesis. Contraction. So, when you contract the water vacuole, it's microfilaments helping that happen. What's cool about the cytoskeleton is that unlike ours, if the cell needs to drastically alter its shape, it can break down the cytoskeleton in one part and reassemble it in another in order to help alter its shape. And that's why when you see like an amoeba move, it can sort of like expand part of its cell membrane. It'll be right here, and then it expands part of its cell membrane and then flows into it. So it's like here, and then it expands it this way and flows into it. And what it's doing is breaking down the cytoskeleton one part and then pushing it out this way in order to extend the cytoplasm. It allows cells to change their shape depending on the situation they're in. And that also helps them aid in movement. The cytoskeleton is a large contributor to the motor proteins. Motor proteins are a bunch of proteins that are put together, and they can produce two basic cellular structures that aid in movement of the cells. That would be the flagella or the cilia. So these are pretty cool. The flagella you can think of almost like the propeller of a boat. So a flagella looks kind of like a tail hanging off of a microorganism, right? Here's my cell, and here's my tail. And when it's time to move, the tail rotates, kind of like a propeller. And that helps propel it kind of in a random direction. Whereas cilia move like the oars on a boat, right? Anyway, whoosh, 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 whoosh. That's how cilia move. Flagella rotate like a propeller, whereas cilia move back and forth like oars on a boat. The end result, of course, is that the cell is able to move. So, and this isn't due to help from the cytoskeleton and the motor proteins that help drive the movement of those structures, the flagella and the cilia. So here's some cytoskeletal slides. And so they've been able to fluorescently dye the thick filaments of the cytoskeleton. So there we go. You can see them all throughout there, these microtubules all throughout the cell. And they're invisible, but they're there and they help provide structure, especially the microtubules that provide structure, and they can provide little tracks on which vesicles move, which might be what we're seeing right here. It looks like something a vesicle could move on, or who knows. And then down here you can see pretty well how they anchor organelles. So here's a mitochondrion, and here's endoplasmic reticulum. And then we have all these little guys, and these are microfilaments and what we call intermediate filaments, which are a thing you don't really need to know. Microfilaments being the little thin guys who can help contract stuff. And then you have these intermediate filaments helping to make this web of network in there, holding things. They're anchored to the organelles to keep the organelles in place. These blue bits are the microtubules existing in there to provide a scaffolding to which the microfilaments and intermediate filaments can attach. And then down here along the bottom we have more microfilaments that are ready for cytokinesis, that are ready to alter the shape of the membrane. They can contract, so it's pretty cool there. So, like I said, a flagella is a long tail-like extension that spins like a propeller, and is specifically for movement only. Sperm are a great example of a cell that has a flagella. Then we can contrast that with cilia, which are shorter, thinner extensions, kind of look like hairs, and they move like oars, and they always occur in multiples. And they can be used in movement, but at the same time, if you have a cell that is stuck in one spot, it can move its cilia to move fluid across the top of the cell. So it can help in moving fluid around. So there's a bunch of cells that line your sinuses that have cilia in them. And normally they are beating their cilia to keep the fluid in your sinuses moving around. However, when you get sick and your sinuses get full, that cilia can be sort of overcome. You can shoot salt water up there to try and kickstart them into action to make your sinuses clear out more quickly. So, kind of neat. So, like I said, they can be used in movement and move fluid across a stationary cell's surface.